Mm-hmm.
It says like the last thing to occur would be the cross bridge cycling occurs. So do we have to have that coupling for the cross bridge cycle to occur?
Yes, yeah, that's how the cross bridge cycle occurs. So the excitation contraction coupling, the whole idea of it is that, like, in order for our muscle to contract, right, the reason it's contracting is it got an action potential, like it got a signal from a nerve. So the excitation contraction coupling is how we get from that action potential coming down the nerve to the actual contraction. So the reason the cross bridge cycle is at the end is that's the contraction part. So the action potential coming in, going through the junction down the T tubules, releasing calcium from the sarcoplasmic reticulum, that all has to happen first. We need calcium in the cytosol in order to do the cross-bridge cycle at all, because otherwise the tropomyosin is covering the myosin binding sites. We can look at a picture if that was a lot of words.
Yeah, it was a lot. But I mean, that was mainly my thing when I was going through it. It said that's when it would occur. And I kind of looked at it and was like, oh, that's a total different thing. But I guess they're not.
Yeah, we look at them as two things, but this is like the setup for the whole thing. I'm going to just zoom the picture. I think you guys can see it, right? So this is showing us the junction. This is showing us the acetylcholine coming out. So that's how we depolarize. This is showing the depolarization going down here, calcium coming out. And calcium is key to this cross-bridge cycle, because if it's not bound to troponin like this doesn't occur, because this pink thread is the tropomyosin. So it would be blocking the myosin binding sites. So we wouldn't be able to cycle. That was a good question, though. Anybody else have things they want to go over, want to take another look at? Want me to do some talking about? Okay. It's quiet. So I'm going to share the document then. As you have questions, maybe we'll check back together every so often to go over things as a group, because I don't want to leave you entirely on your own. There will be a key to this. I have not written the key yet. So if you have questions immediately, it's best to ask questions about these. Okay. So feel free to skip around. So like kind of skim the questions and see where you think you need to put in a little extra work and focus there. What's the time? Okay, it's 51. So we have like 40-ish minutes of class. So I'm going to put you into groups, but I'm going to bring you back like every 10 minutes. And I assume everyone will have a question. Someone will have a question 10 minutes from now. I'm pretty positive. Okay. So. That seems good. Oop. Still sharing my screen. All right. Anybody got a question yet, or a thing they want to go over after looking at that worksheet? No, I just disrupted everyone by kicking you out of those breakout rooms.
Can you go over the first question?
Sure. So what the first question is doing, right? It says to do a flow chart of the nervous system. So we're using these terms: afferent, parasympathetic, sympathetic, efferent, sensory, motor, somatic, skeletal muscle, smooth muscle, receptors, CNS, PNS. So basically what you should be doing here is pulling out pieces of this chart. Am I screen sharing? I am screen sharing. Cool. And the reason we're pulling this out is that this kind of holds together like all the information for this unit practically is somehow related to this chart. So you should always be thinking as you're studying, like, where in this chart are we? And just in general, this unit is set up with kind of a lot of compare and contrast things because of the branching form of this chart. So one thing that we'd be comparing and contrasting, right, is we have sensory information coming in. So you'd put afferent and sensory on kind of this line going up. Efferent and motor would go on this line coming out. You'd have CNS here in the middle. Your receptors are going to be at the opposite end of the sensory. So go like receptors, sensory, CNS, motor, efferent. And then we're going to have a bunch of splits. So you're going to have somatic and autonomic. I realize an autonomic isn't on your list. Probably should have put it on that list, but you would have the two divisions here, parasympathetic versus sympathetic. And then we have some examples of effector organs. So I think I gave you skeletal muscle and smooth muscle. So at any point here, right, we're always comparing and contrasting, right, our receptors and neurotransmitters in the parasympathetic versus the sympathetic versus the somatic. We're comparing like the types of connections. Let's say that the nervous pathways we'll see in the sympathetic, right? Like we'll go through the sympathetic ganglion in the sympathetic division versus we won't in the parasympathetic. So I feel like this is a really good place to start with your studying, because I think you should be able to link almost everything back to this, the exception being some of the endocrine stuff. Although I will note for you here, one of the places we're going here is glands. And we have seen how our neurons are involved with our endocrine function because of that connection between the hypothalamus and the pituitary. Okay.
Could you go over question 8 with the corticotropin releasing hormone?
Absolutely. Let me pull up that question exactly. Question 8. Okay. I'm gonna flow through. So that's coming from the endocrine section here. And what the ED's getting at really is this idea of hypothalamic pituitary axes, and basically how tropic hormones work. Okay. So we had a couple of places we looked at these because we looked at the hypothalamus and the anterior pituitary itself. And then we also looked at cortisol independently. So we kind of want to link these topics together. There we go. Okay. So the question is asking about corticotropic releasing hormone, adrenocorticotropic hormone, and cortisol. So a good place to look at the first is we can look at this chart or this chart, kind of the same. In your version of this chart, I wrote up at the top what would be coming from the hypothalamus. I like this one a little better just because it has the names written out, but this one is nice and color coded. So what we'd be circling there for number 8 is we're talking about this pathway. So we're talking about the fact that the hypothalamus is going to release CRH, that's the corticotropin releasing hormone. So that's a tropic hormone, right? A tropic hormone being one that affects the release of other hormones. It goes to the anterior pituitary, and we want to remember as we're doing that, the anterior pituitary is special because it has this portal system. So the anterior pituitary, there are nerves in the hypothalamus. We have these 2 capillary beds linked by a portal vein in the infundibulum. And then we have an actual endocrine cell in the anterior pituitary. It's not true for the posterior pituitary. The posterior pituitary is just neurohormones. Okay. So we're releasing our tropic hormone from the hypothalamus. It goes through the portal system to the anterior pituitary, where we release another tropic hormone. This one's still a tropic hormone because it's also affecting the release of another hormone, which is cortisol from the adrenal cortex. And the other fact that we might want to add on to this, this one. Okay, is this idea of feedback. So this is showing us that flow chart in another way, CRH to ACTH to cortisol. But on this chart, we can also see that cortisol is giving us a form of long loop negative feedback. And the long loop negative feedback is when this like final hormone loops back and turns off one of these upper parts of the pathway. So cortisol is looping back, and it stops this loop by suppressing the release of corticotropic releasing hormone and adrenocorticotropic hormone. So the cortisol secretion should turn off this signal eventually so that you don't have completely runaway cortisol levels. The other thing we might want to know, right, is just making sure we know what the adrenal cortex is, right, versus the medulla, because the adrenal cortex is here in this endocrine section, but we remember that the medulla was this inside part with the chromaffin cells releasing epinephrine and norepinephrine. So that was part of our sympathetic nervous system. So we're talking about the outside as endocrine, inside we're thinking about more as nervous. Okay, I'll give you a little more time to work through stuff and come up with some more questions for me. Sending you back. All right. Somebody want to pick another question?
Number 3.
Okay, cool. Pull that up.
We just go.
Sorry, you cut out for a second. Can you hear me? Oop. I'm going to send a message out. Can you guys hear me?
Yeah.
Oh, you can. Okay, cool. Okay. So number 3. Okay, so when we're talking about number 3, let me pull up the slide we should be looking at. So we're thinking about the parasympathetic neurons versus the skeletal neurons. So we see those in kind of 2 places, right? So one thing that we're looking at for parasympathetics, we have pathways like this. So our effector organs for this are going to be kind of the same as our effector organs for the sympathetics. So they're going to be smooth muscle cells, they're going to be cardiac muscle, some glands. But specifically what we're usually going to see are muscarinic cholinergic receptors. I'm going to just have us take a glance at what that receptor looks like, if I can find the picture. Nope. Okay. Maybe we don't have a picture, but just a reminder that well, they're receiving acetylcholine, and they're slightly slower than the nicotinic cholinergic receptors. I can't find the picture. But for the skeletal muscle, we're talking, You found one somewhere? I think there is a picture somewhere. But basically, it's about the 2 types of cholinergic receptors. The parasympathetics, we're going to see muscarinic, which have more varied responses, whereas the nicotinic cholinergic receptors that we see in skeletal muscle are basically just ion channels that open. Any other questions? Burning questions? No. Before I forget, so I've been working on this key while you guys are in your breakout rooms. So we're probably doing similar things. I'm just gonna make sure that I share this key with you so it's in progress because I am still writing it. But I'll make sure you have the location for that. Where's my chat? There's the chat. Okay. So you should be able to see this. And I myself have been kind of jumping around. Okay. Put you back in your rooms. Okay, the sharing on that document should be fixed. Just gonna send that link one more time. And I'm gonna stick around for questions. One thing to note is I've been inputting your lab grades. I'm not finished with that, but if you have notifications turned on, expect to see many coming through as I'm inputting all of those. If you see errors in there or questions, there shouldn't be errors, but do let me know if you have a question or a problem with what's going in there, and we can sort it out.
I have a question about number, like the second part of 9.
Okay.
Asks, what other responses could you expect to see at the same time if cortisol is released?
Yeah. So when we're talking about cortisol, one of the cool things about it is that it's part of this, cool, right? It's part of this general adaptation syndrome, which is like a very characteristic response to stress. So the basics of that is that the whole thing is going to be like a fight or flight response. So we're going to see that cortisol, when it gets released, there are other hormones involved or other things involved with that fight or flight response that are going to be going on at the same time. So that's this slide, pulling it up. So when we're releasing cortisol, we're also going to expect to see increased sympathetic activity, which is going to come along with increased epinephrine secretion. There are some other things we're going to see also kind of released at the same time, ADH. So that's one of our hormones from our posterior pituitary, angiotensin 2. But the whole goal is this fight or flight response. So anything we associate with that is going to be released at the same time. Our goal is to mobilize energy stores. So to start breaking down glycogen, kind of like when we were talking about the post-absorptive state. And we're trying to maintain blood pressure because we're going to try to keep all our systems going in order to address whatever's stressing us out.
For the tropic hormone, is it inhibited by negative feedback loops?
Yep.
Okay.
Yeah, they're inhibited by negative feedback loops. And we have two kinds of negative feedback loops that we think about with the tropic hormones. We have the short loop and the long loop. The short loop is when like the hormone released by the anterior pituitary doubles back immediately to the hypothalamus. It's short because they're right next to each other. They're like right there in the brain. Whereas the long loop is when we had that anterior pituitary go out to an endocrine organ in the body, and then that final hormone loops back, that's long loop. But they're both forms of negative feedback.
Can you go over 15?
Mm-hmm. Sure. Okay. Gonna scroll out of here. So when we're talking about excitation contraction coupling, that's kind of like what Amanda was asking about at the very beginning of class. So we're looking at after the cross-bridge cycle. So we're looking at this stuff. Okay. So the first part, why calcium is released. the reason we're releasing this calcium essentially is in response to an action potential. So it's being released because we had a motor neuron releasing acetylcholine in the neuromuscular junction. We had these nicotinic cholinergic receptors that created a depolarization in our muscle cell.
Can you share your screen?
Oh,
Were you looking at
oh,
something?
sorry. I am looking at the screen. Thank you. There we go. Okay. So we have this motor neuron releasing acetylcholine. Is always depolarizes a muscle cell. So that's important to remember about our somatic nervous system, is that we're not going to see inhibition here. When we have an action potential in our motor neuron, we're going to fire it off. So we see this depolarization here running down the T-tubule, communicating to the sarcoplasmic reticulum, and that's when calcium is released. So short answer, calcium is released because we got an action potential from the motor neuron. Long answer, we could go through all these steps. What effect it has on the cross-bridge cycle is that when this calcium gets released, we need that calcium for the cross-bridge cycle to even occur. So the effect is basically that it starts the cross-bridge cycle, because without this action potential, right, like we only want to contract our muscles when we're getting a signal to do so. So we don't have the cross-bridge cycle going without this action potential. So when calcium is there, that's like the trigger that exposes these myosin binding sites, allows myosin and actin to bind to each other, and allows us to enter that cycle. That's why we call it the excitation contraction coupling, is that it's linking these two things together, linking together the excitation, which is that depolarization, that action potential from the nervous system, and the contraction, which is the cross-bridge cycle and that sliding filament model, the muscular side with the sarcomeres.
Is there going to be anything on the exam that, or not on the exam that we like went over in class? Like anything specifically?
Yeah, that's stuff from the first day with the central nervous system. We're not going to do. That was just, I wanted to make sure that you knew what the blood-brain barrier was, because you'll hear about it in future, like when you talk about medications and stuff, but we're not going to test on that. And we're not going to test on any of the packet that we didn't get to. We only had a little bit left of the skeletal muscle packet anyway, and most of it was covered in physiology lab. So we were talking about the last slide we did, I don't know what number it is, but it was that there's an optimal length for sarcomeres. And we didn't talk about why there's an optimal length or how that works. So if that were to appear at all, I would guide you through it, and it would be an extra credit question.
Can you go over question 12 as well, please?
12? Sure. Okay. So this is talking about the different parts of the sarcomere and how we name them. Pull that up. Okay, you guys can see my screen this time, right? I hope.
Yeah.
Okay, cool. So when our sarcomere shortens, right, the thing about this sliding filament model, the reason this was a cool thing that they discovered, right, is that none of our proteins are actually getting smaller. They're just sliding against each other. I'm sorry, this image is formatted weird, but I think the image in your slides is okay, the one that you have. So the reason the A band is the same size is that the A band is just this part where it's anywhere we have myosin. So the A band is never going to shrink because the myosin never shrinks. All we have happen is that the actin slides over and covers up more of the myosin. And this is why we see the H band or the H zone shortening, because the H zone was where we had myosin, but not actin. But for the A band, it didn't matter whether it was covered by actin or not. It's just the length of the myosin, and the length of the myosin always stays the same. So another thing, just looking at this image, since we're here, just note that the I band is also shortening. So anywhere where we're talking about having just one of the two, H zone just myosin, I band just actin, both of those are shrinking as we create more and more overlap.
Can we go over where tropic hormones are produced?
Yeah. Okay. So tropic hormones are going to be produced in multiple places, but primarily what we're thinking about are the hypothalamus and the anterior pituitary. And that's because of our definition of a tropic hormone, which is just one hormone that affects the release of another hormone. So like, scrolling back to this chart again. Okay. So when we were looking at this, all of these hypothalamic hormones count as tropic hormones because they all have an effect on the production of a hormone in the anterior pituitary. So right, PRH is increasing prolactin secretion. PIH is decreasing it. TRH is increasing TSH secretion, and so on. That's why we see all these pluses and minuses. So all of these hormones from the hypothalamus being released by that nervous tissue, they count as tropic. Then when we come to the anterior pituitary, we can see, right, our better examples would be these ones. These are more clear. But these are going to be tropic hormones as well. Like TSH is going to the thyroid gland, which is then going to release thyroid hormones. ACTH is going to the adrenal cortex. It's releasing cortisol. So these would also be tropic hormones because they're affecting the release of more hormones from these other organs. And they're not all drawn out, but we're really concentrating, if you're concentrating on pathways, I think I said this at least once before, but I'll say it again. If I ask you about some of these other pathways that we haven't talked about in detail, I'll really be very explicit and kind of explain these to you. The two I would expect you to be familiar with are this TRH, TSH, thyroid, CRH, ACTH, cortisol. And that's because we went through the thyroid gland separately and we went through the adrenal cortex separately. We haven't really gone through any of these other systems yet. We'll get to them when we do like reproductive stuff. So while this chart is overwhelming, just kind of focus on these two.
I guess I have one more question. Could we go over alpha and beta receptors?
Yeah. So both of them are the alpha and the beta adrenergic receptors responding to like norepinephrine, epinephrine. So we're associating them with the sympathetic nervous system. I'm talking while looking for the slide. But basically the big difference between them is going to be cyclic AMP. So we have all these various different kinds, right? Whenever there are Greek letters, I start to panic a little bit, but it's fine. It's not so bad, right? So we have our alpha receptors and our beta receptors. And the big difference, it's very convenient. Beta receptors always activate cyclic AMP. So we can see that here. So that would always be my first step if I was trying to identify what type of receptor I'm looking at. Look for cyclic AMP. If it's activated, you're looking at a beta receptor. The confusing little bit can be that our alpha-2 receptors, you also might see cyclic AMP, but it would be inhibited. So otherwise, you could also think about the relative affinities for norepinephrine versus epinephrine. And we'll talk a little bit about this when we get to the cardiovascular system. This will come back up again, but we'll focus on that more when we get there. Let's just get the basics down for now. Just scrolling back or scrolling forward, I guess. A thing to note about these adrenergic receptors is that they're going to be G protein coupled receptors. So if you were looking at one kind of zoomed in, you'd expect to see something like this. You'd see norepinephrine or epinephrine coming in, linking up to a G protein, and then doing something. This one doesn't include cyclic AMP anywhere, so this would be an alpha-1 receptor. And then when we look at this image, this is from your textbook, but it's kind of horrible. So I did explain it in class. But this one's trying to show you the difference between an alpha-2 receptor and a beta receptor. And they don't make it at all obvious, but there's a minus on the side with the alpha-2 receptor, which is telling you that it's inhibiting this, versus the beta receptor has a plus on this side. So it's exciting this pathway. So basically what they're trying to get to here is that the alpha-2 receptor kind of like fights against the beta receptors. I suspect you're all just hanging around in case someone else asks a useful question. Is that right?
For the last thing for me, can you go over number 5, the difference between the neuroeffector junction of the autonomic and neuromuscular junction of the somatic?
Yeah, sure. I don't know why I keep unsharing this screen. I should probably just keep it up. So there are a couple differences and a couple different terms we want to associate with each. And part of why I was asking this question is that I think we covered them on different days. So it's helpful to compare and contrast them now to realize that they're one of these kind of compare and contrast topics. So the neuromuscular junction is what we've been talking about with skeletal muscle. So that's a helpful thing to realize, is that all this stuff with the excitation contraction coupling, the cross-bridge cycle, so that's all coming from the neuromuscular junction. So anything we talked about as we were talking about that excitation contraction coupling is going to be part of the neuromuscular junction, but not the neuroeffector junction. So I have up here the neuroeffector junction, so we can highlight some differences here. So one thing, I'd like to take a look at the varicosity. Okay, so when we're talking about neuroeffector junction, we're talking about autonomic. So this effector organ might be a gland, it might be skeletal muscle, any of these things. So one difference is in where these neurotransmitters are getting released. So in the neuroeffector junction, we see these structures called varicosities. So we see several swellings down the end of essentially these axon terminals, which is weird. We haven't seen that before. We don't see that in any of our other examples. So varicosities in our neuroeffector junctions versus, be good if I could flip them. I'll kind of split screen this. Hold on. The comparable structure in the neuromuscular junction is the terminal bouton. This is not going to be great. I'll just click back and forth between these. Are you guys seeing my whole screen? I want to make sure. Yeah, you're seeing screen too. Okay. Okay, so here we had our varicosities, these kind of swellings, versus here at the neuromuscular junction, we just have this one terminal bouton. So another thing we might think about here are about the receptors that we're seeing. Actually, that's not that helpful. The receptors aren't that helpful. The effect of the neuroeffector junction is going to be kind of more general because we don't see it linked up as tightly at the neuromuscular junction. We're also going to see a bunch of specific words with the neuromuscular junction that we don't talk about at the neuroeffector junction. So like at the neuromuscular junction, we talk about, I need to make this way bigger. Okay. So at the neuromuscular junction, we have this motor end plate. We talk about an action potential, depolarization. We talk about an end plate potential. Those are all with the somatic nervous system, with this skeletal muscle, with this neuromuscular junction. Versus in the neuroeffector junction, we're going to have a lot of different types of responses, but they're going to be much more general. Like we don't have this little pocket for the varicosities, right? They're just kind of over the surface, and we'll see several of them over the surface, versus we have this motor end plate that creates a really specific kind of one-to-one junction here between the motor neuron and the skeletal muscle cell. Yeah. Okay. So yeah, the main differences I'd focus on are the varicosities versus the terminal bouton, and then the fact that the neuroeffector junctions have a kind of general effect, whereas the skeletal muscle neuromuscular junctions are really specific. The thing I was saying about the receptors is that it's true that we'll see muscarinic, cholinergic, and adrenergic receptors in the neuroeffector junctions. And we're always going to see nicotinic, cholinergic receptors in the neuromuscular junction. But if I were going to ask you about the muscarinic, cholinergic, and adrenergic receptors, I think I'd ask you about them in a different context than this context, because this is kind of putting a couple too many steps together for one question on the exam.